Okay, let's get started then. So welcome back everyone to the Berkeley Superfund programs, uh, pretty informal Monday, once a month, get to know you, learn more about PFAS discussion group. And like we said, these are meant to be some shorter talks to kind of inspire us and to teach us about what other people in the community of practice are up to. And today we get to hear from John Jacob, who's the state toxicologist for the state of Hawaii's Department of Health Hazard Evaluation and Emergency Response Office. John has a PhD in pharmaceutical science from the University of Hawaii and has been very much involved in some of these PFAS issues, especially as they're relating to the food supply. And that's a growing important topic that I think this group has expressed some interest in. So without uh, any further ado, let me turn it over to John, but let me just say quickly, if you have questions during the talk or at the end of the talk, please put them into the chat so that we can keep record of them. And it's a little hard to keep track of all the raised hands. So that's probably the best way to do it. John, thank you for your time. We're looking forward to hearing from you.
Awesome. Thank you. Thank you so much for having me. Good morning, everyone. So today I'll be talking about one of our ongoing projects entitled PFAS Uptake in Food Crops from Compost and Domestic Wastewater, a Field Study. So with this project, we're trying to answer two main questions. And as some of you know, Hawaii has limited drinking water for drinking and for irrigation, even though we're surrounded by the biggest water body on the planet. So the question we're asking is, R1 water, that is recycled, disinfected water, safe for agriculture use and in terms of PFAS toxicity? If yes, that'll give us more fresh water for drinking and will also help the community and the state's agriculture industry. So currently, most of the R1 water is only used for irrigating lawns and golf courses. So the second question we're trying to answer is concerning the fact that Hawaii also has a waste or landfill problem because we have limited land compared to the other states. And there have been community initiatives to use more compostable servicewares. So the question we're trying to address here is compost made from PFAS containing compostable serviceware safe in terms of PFAS uptake in agriculture crops? So previous studies, as here you can kind of broadly give a summary of previous studies up until 2021, where here they saw the summary of the name of the authors and the vegetation where they looked for PFAS uptake and the compartment where they detected PFAS, the amount bio availability or bio accessibility, and the range of PFAS they looked. So as you can see, the lowest is in terms of carbon chain is C4 and the highest is C14, which means they looked only at long chain and short chain and not ultra shorts. So from other studies, along with the same study that I discussed before, they kind of summarize that perfluoroethanoate and perfluoropropanate are the most common ultra shorts involved in plant uptake. And that could be mostly because they're water soluble, so they can be easily taken up by the plant. Also, they may be a lot more available because they can be breaking down products of long or short chain PFAS. And the state of Hawaii already have environmental action levels for both the compounds. And some may argue that ultra shorts are comparatively less toxic, so it's not a big deal, but it's in higher concentration, especially when it comes to plant, because of the fact of accumulation, they can be still very toxic. So considering all that, the state of Hawaii is mostly concerned with total PFAS risk. So that is the sum of primary terminal PFAS. Those are the most commonly detected or looked for PFAS. Then secondary terminal PFAS, those are PFAS that may have gone undetected because of other precursor compounds attached to the PFAS molecule. then excess fluorine. So that's the ones that doesn't come in any of these categories, and they also impart risk or toxicity. Or in other simple words, we're interested in risk, the total risk associated with long chain, short chain, and ultra shorts. But in terms, if you want to define it in terms of methodology, we're looking for pre-TOPS, that is total organic precursor, post-TOPS, so after the TOPS method, and total organic fluorine that detects the excess fluorine, excess organic fluorine. So it's like calculating the total calories in a burger. You want to look at the contribution made by the major contributors. In this example is the patty, the bun, and the minor contributors are the sauce, cheese, and veggies. Now just replace the calories with PFAS risk. So the major contributors here, mostly the long and short chain. And if you ignore the minor contributors, that's the organic excess organic fluorine, you're still missing out 33.1% of the total risk. So that's why we're interested in the whole package, and that's where we're coming from. And from our previous study where we looked at wastewater treatment plant effluent, we found that the major PFAS composition comprised of mostly of ultra shorts, as you can see here. And even though they're 100 to 1,000 times less toxic because of their higher concentrations, they contribute to a higher risk or toxicity. It's like the saying, concentration makes the poison. So with all that said, our ongoing study will first determine the baseline levels of PFAS in the food wastes, servicewares, municipal water. We're assuming municipal water to have little or no PFAS. And that's why we're doing the baselines, and R1 water, and the 3 composts that I'll be talking about in a bit. And the vegetation that we're looking at or exploring in the study are the fruits. So in the study, tomatoes, leafy vegetables, we're using kale. Fruits, we're using carrots. And the compost, the first study group is with the serviceware. So it'll contain green waste, food waste, serviceware, and irrigated with municipal water. And the second study group where we're looking at R1 irrigation water, the compost comprises of green waste, food waste, and R1 water instead of municipal water. And we have a control group that is made from green waste, food waste, and municipal water. And this will be used to compare the other 2 study groups. We're also interested in knowing which PFAS are accumulating at which part of the plant. That gives us an idea of which part of the plant is more or less toxic in terms of PFAS. So we'll be testing different parts of the plant separately to identify the risk and or safest part of the plant. So with the results, we're planning to make decisions and legislations, and there are 3 hypotheses in terms of R1 water and serviceware. That is, it is safe for all consumable crops. It's safe for some, but not all. Not safe for any consumable crops, and we should only use them for non-consumable crops such as hemp. And we anticipate to finish the study by the end of next year. And with that, I'll conclude my talk, and I'll take any questions if anyone has any. Thank you.
Great. Okay. Well, yeah. Thanks, John. Those of you who have questions, please type them into the chat. I wanted to start with a question, and then hopefully by then someone else will have raised their hand, and so on and so forth. So, John, one of my questions here is that it seems like the composition of compost is not uniform. Like food waste is going to vary from location to location, and that it's also likely to be changing over time as the manufacturers alter the kinds of products. So in your study design, how do you assure that you have like a representative compost? And if you and if this were to be repeated later, how would we know that, like how do you characterize it, and what kind of information will you have to report on it?
Good question. So the food waste that we're taking is from a single or two, I think one or two sources. One is from school, the other one is from a hotel. So we're anticipating it to be the same turnout because it's a known source and it's not randomly collected over the municipality or whatever. So it's from a single school and a hotel. That's how we know it can be representable.
Okay, good. And yeah, and then the soils, you must be growing in more than just compost. Like in addition to compost, is there soil, or do the plants grow in pure 100% compost? And is that what would happen on a field?
It's going to be 100% compost.
So it's like a worst case scenario that you're testing at this point.
Correct. Correct.
Okay,
And we have an
good.
expert, compost expert, working with us on that matter.
And so the plants I mean, I can imagine they're happy growing in that rich organic matter.
That's the idea.
Yeah,
Because we know the soil are not that, we already did the studies. So soil, we know that they don't have much PFAS or no PFAS at all. So it's no point of adding that. We just want to straight get to our question.
okay. And then you and your colleagues have kind of come to this conclusion that the ultra shorts are going to be the largest risks because of some assumptions you've made about, I guess, toxicity, like projecting forward the toxicity. What do you think are the most sensitive aspects of your assumptions? Like where is there more research needed?
It's mostly, so we're still doing, I personally am still working on understanding short chains, uh, particularly because, long chain, short chain, they can break down.
Hmm.
So ultimately, the way I see it, if the world decides to stop manufacturing PFAS cold turkey, along the line, maybe, I don't know, 20 or 50 years along the line, we're just still going to have a lot of ultra shorts.
Yeah,
That's going to be the next issue. So we're just getting ahead of the game.
right,
And since our one water, like I showed the data, is mostly um, ultra shorts that we're finding. So we're mainly looking at the safety aspect in terms of ultra shorts
right,
along with the other, but our priority is ultra shorts, or our question is basically addressing the ultra shorts.
great. Okay, so Narissa has a question. Narissa, why don't you ask your question?
Thanks. I actually have a couple questions. One is about, I guess, rainwater and the ability for the PFASs to move in the soil compared to pots. It looked like maybe you were growing in containers. And then the other one is, I mean, 100% compost would be worst case scenario in terms of PFAS loading to the plant, but also the composition of the compost, because it's such highly organic material. Wouldn't the compost be maybe holding on to the PFASs differently than a less organic mixture would be?
That's a good question. I will have to check with our compost experts on that. But we're doing baselines on everything, so we should get a representative, at least an idea of how that will work.
Mhm. Kathleen had a question.
I was just asking about what you know about the municipal water. Do you know it to be low in ultra shorts?
Yeah, we know they have a little bit, but it's very negligible, and it's ultra shorts. But because it's a different site, we still need to do the baseline regardless.
Good. And Michelle has a question about the toxicology, which is more up your alley. Michelle?
Yeah, hi. We're working on some assessments of ultra shorts in my lab right now, and I was just curious, what type of toxicology data does the state of, excuse me, Hawaii consider in terms of toxicity? Like, are they willing to look at NAMs, like in vitro data or even in silico data, or is it more traditional in vivo rodent testing? What's your sense of that overall regulatory or decision-making
So
landscape?
talking to Roger and the folks in Australia, they think animal studies are more reliable.
Sure.
So that would be ideal, but we have limited capacity to do large scale animal studies. But so that's one of the reasons we're looking at risk than toxicity, direct toxicity.
So risk just in terms of the summation that you're talking about and not being concerned about the specific tox tests with these chemicals per se.
Yes and no. Test testing, we still require, or we still recommend all three tests, pre-tops, post-tops, and tops. But within that, we have a risk calculator on our website that kind of gives us the risk if we put in the values and detected levels of PFAS, and there's a lot more parameters to it. So that gives us an estimation. It's more theoretical than very direct value. So, and also because Hawaii doesn't have a big concern because we don't have a lot of manufacturing industries. We're mostly worried about the military, where they use a triple F form, and a lot of the hotel mass scale washing, linen and cloth washing, a lot of the cloths are covered in PFAS for stain resistant and whatnot. So those are our primary locations. So compared to the estate, we don't have a lot of PFAS generators.
Okay, thanks.
So I had a question about an analytical question about the top assay. It seems like it can be a little challenging to run it on an organic rich material like biosolids. And I also am not aware of top being used on fruits and vegetables and plant tissue yet. So who's dealing with that issue, and where are you with those?
That's a very good question. I knew someone was going to ask me that. So the current method, what's available is mostly for fish tissue. So I myself, Roger, and some of the labs are working on, or more of exploring different techniques that will allow us to test a representable sample from plant tissue to detect PFAS. So it's long story. Well, the short answer, or the easy answer, is it's still work under process.
And are you using like a solvent extraction, or like are you freeze drying the samples and then extracting them? Like how do you deal with these matrices?
We are kind of moving towards freeze drying. So that'll be ideal, but a lot of the labs doesn't do that. Because a lot of the commercial lab, they stick with their protocols. They don't like exploring new. And that's not all. Some are like that. So the ones we are working with, they are trying to freeze dry was one method. The other one is liquid nitrogen, and freezing down and crushing it and using a subsample that's representable.
It's very interesting. Great. Other people on the call have anything they'd like to discuss on this issue? Okay. Well, you all have, I think, John's contact information. He's part of the community of practice if something comes up and you'd like to discuss this more. But I think that this was a nice introduction to a topic that is likely to become more important. And at the beginning, I put the link to the New York Times article about the biosolids that was in that story earlier last month. Martin, did you want to say something? I see you come off mute there.
Yeah, I'm here. So John, a quick question. Are the ultra shorts toxic to the plants. What worried me about, if you've got high concentrations in compost, I mean, things like trifluoroacetic acid would surely be toxic.
Right. So a lot of the literature that I've read, they saying that it doesn't impose maybe at very high concentration, but the study summary they directly doesn't impose any noticeable toxicity to the plant, directly to the plant. So we're going with that for this study. Of course, we'll find out if it's toxic because we're using a way higher concentration than these other tests have done. So that's almost in this exploratory state.
Okay, thanks. Great talk.
Thank you.
Great. Okay. Well, these are just meant to be a little snack before lunch, these meetings. So great to hear from everyone. And we'll see you, I guess, in a month when we get to the next one.
Okay. All right. Thank you, everyone.
Thanks, John.
John,